Stevens Institute of Technology. As a sociocultural anthropologist, Dr. Benezra's academic focus lies at the intersection of anthropology and microbiology. Her work, based on field studies in Bangladesh titled Anthrobiota, Co-Evolving an Anthropology of Microbes, is the very first ethnography of the microbiome. She is a pioneer in the exploration of how combining microbial ecology and social science could benefit global health. And she will talk to us today about who decides what healthy is, studying microbiomes across disciplines.
Thank you so much. Are my slides good?
Yeah, you were great. Thank you.
Great. All right. Thank you so much for that introduction. I'm so happy to be following Dr. D'Amato because her work is wonderful and a perfect segue to what I want to talk about. And thank you to Sean and Catherine for inviting me to be part of this symposium. This talk is going to be much different than most of the talks that you've heard today. Well, no, it's going to absolutely be totally different than everything you've heard today because I'm a sociocultural anthropologist by training. That's what my PhD is in. I'm really glad to be here though, and I think it says something very significant that I'm a part of this symposium, and I'm really grateful for that. So hopefully this will all make sense to you all. And anthropologists typically read their papers, and so that's what I'm going to do. I'm going to be reading. So hopefully that's not too jarring. It's very different than a scientific presentation of data and how scientists usually talk about their work. But here we go. Dhaka, Bangladesh looks lovely in the earliest hours of the day. When the sun rises over the bamboo scaffolding of half-finished high-rises, the people are quiet and birds and insects fill the sky. The morning light is beautifully tinged with exhaust, pollution, and dust. Within minutes, it becomes intense, bright and hot, the UV index often forecasted at a maximum 10. The sun starts early here. In Mirpur, one of the most densely populated localities and the most densely populated place on Earth, mothers rise in one-room shacks, chase mice from cooking pots, boil water, and prepare food for the day. 8,000 miles and 11 hours later in St. Louis, Missouri, scientists will arrive at the Gordon Lab, sacrifice mice, segment intestines, and prepare cells for sequencing. The microbes in the guts of these mice are from the guts of Bengali babies. They traveled across the world in test tubes, in fecal samples collected by mothers and packed in picnic coolers in the stifling Dhaka sun. These microbes traveled by airplane on flights that cost more than the yearly income of most Bangladeshi families. They arrive at the Center for Genome Sciences and Systems Biology at Washington University, a gleaming multi-million-dollar facility with several sequencers, a mass spectrometer, and a computer cluster capable of analyzing trillions of reads. These microbes will be injected down the throats of black 6 mice. I think many people were talking about germ-free mice in their studies, so that's what the Gordon Lab has as well. There's more here than a predictable cross-cultural comparison between a globally southern slum and a globally northern scientific laboratory. These spaces are more than geography, more than poverty, and more than notions of power, histories of colonization, more than practices of living and knowing. They don't just overlap at the boundaries of the body. Between the Gordon Lab and Mirpur, the social and biological become equally important to understanding human microbial relationships. Based on a collaborative partnership with scientists working on the microbiome, my research follows commensal microbes in the human gut as objects around which scientific and anthropological imperatives are emerging. I conducted fieldwork at the Gordon Lab at Washington University School of Medicine, where scientists were studying the interrelationships between diet and microbial community structure and function, and whether differences in human gut microbial ecology affect predisposition to malnutrition in children. I also worked at the lab's field site in Dhaka, where Bangladeshi mothers and children were enrolled in a microbiome and malnutrition study. There I worked with scientists and researchers at the International Center for Diarrheal Disease Research, known as ICDDRB. The Gordon Lab was attempting to develop a translational research pipeline in order to alter the microbiota of those suffering from metabolic disorders and nutritional deficiencies. I followed microbes to homes in Dhaka, where microbes were conceptually non-existent and practically a matter of life and death. As lab members studied the relationships between gut microbes and malnutrition, I explored how an anthropologist could become a member of a scientific team and contribute ethnographic data, while asking biological scientists to make space in research design, experimentation, and implementation for social science expertise and thinking. The sort of whole idea was to make the ethical, legal, and social implications of the science not an afterthought to the scientific work, but part of the research design. In our perspective piece in PNAS in 2012, Dr. Gordon, Joe DiStefano, and I created what we called an anthropology of microbes. We wrote, quote, "The development of new methods for understanding the microbial world provides an opportunity to reevaluate the way that we view our human, biological, and cultural diversity," end quote. We began to consider how the natural and social sciences might use studies of the microbiome to jointly engage pressing public health problems like malnutrition. Sociocultural anthropologists study the complex cultural lives of people, their decisions, their social practices, and relationships. We use the methodology of ethnography to ask questions about people in the world. We do what we call fieldwork. The ethno part is observing and talking to people who are going about their everyday lives as they experience the commonplace and the extraordinary. The graphy part is when we take all that information and we analyze it and write about it. That's me on the right doing ethnography, and my fieldworking partner, Joe DiStefano, on the left. Our work is primarily qualitative. We don't produce clean, noise-free data, and our sample sizes are usually tiny. For instance, I talked to 9 families during my fieldwork in Bangladesh. It's a very small N. Sociocultural anthropologists don't like to make generalized conclusions about cultures, societies, or groups of people, but ethnography provides insights into what life is actually like for people on the ground, giving important details that might not be visible in microbial genomics, for example. It can shed light on the transnational and local realities of people who are embedded in global scientific research and health programs. It can show why health interventions don't work, why they aren't practical or implementable in real life. As an anthropologist, what I love about the microbiome is that it brings together social intimacies with our biological selves, as Dr. Germano was talking about. All of the things about who we live with, where we live, what we eat, all determine what our microbial populations are. They're affected by how and where we're born, what food we eat, who we live with, who we love, and those microbes are digesting our food, training our immune systems, interacting with our states of health and illness, moods, and behaviors. We know that new metagenomic technologies are revealing the impact microbial communities have on human health and are changing ideas about biology and what the human even is. In turn, microbial concerns are emerging for the social sciences, humanities, and philosophy. In my subsequent research, I've continued to develop an anthropology of microbes, drawing together the collaborative capacities of ethnography to actively contribute to scientific projects, partnering with scientists to help develop more holistic, analytical, and methodological frameworks with the goal of understanding the embodiments of biology within social dynamics. These transdisciplinary collaborations are not easy, even if both parties are in the partnership come to the table with open minds, willingness, and generosity. We all know that each of our own disciplines has a particular language, a specific way of conducting research, and even a special way of writing papers. We never really learn how to talk to other kinds of scholars or how to work together. I'll say, just as an aside, Dr. Gordon was probably the most magnanimous and generous collaborator I could have hoped for, and still, in the end, our collaboration did not succeed. I worked collaboratively with the Gordon Lab for about 7 years. For 14 months, I was actually in the lab, conducting my independent research and also serving as an anthropologist in the employ of the lab. I was thinking about how accounting for microbes changes anthropology and how anthropology can collaborate with scientists accounting for microbes. The Gordon Lab scientists sometimes talked about humans as superorganisms, a composite of human and microbial cells, an amalgamation of human and microbial attributes. So I'm sure you all know all of these statistics. I know that more recent data shows that there are 46 million microbial genes in the gut alone. Early estimates said 2 million microbial genes in humans. Obviously, this is really interesting for an anthropologist to think about what humans are if we're actually made up of all these different organisms. At the Gordon Lab, a lot of the Gordon Lab experiments were looking at the gut microbes of babies in Bangladesh and Malawi when I was there. who were suffering from malnutrition, trying to figure out the connections between bodily bacteria, food, and illness. They enrolled mothers and babies in the Microbiome Discovery Project, gave them free healthcare and medicine, and took periodic samples of blood, urine, and lots and lots and lots and lots of feces. Meanwhile, I spoke to these mothers, the Bengali field researchers and scientists, and explored ways to reconcile the scale differences between the lab, the intimate biosocial practices of Bangladeshis, and the looming structural violence of poverty, poor sanitation, and infrastructure. I also talked to the Gordon Lab PhD students, postdocs, and technicians to find out how they were thinking about the work they did, as well as their own relationships to their microbiota. So I asked then, and I continue to ask, what's at stake within these newly understood human microbial relationships? How can we address the bioethical issues? Who owns your microbes? Why are certain bodies, especially those from historically vulnerable populations, the focus sometimes of microbiota sampling? What does it do when science talks uncritically of people of different races having different microbiomes? How can we make sure the study subjects are heard and benefit from the research? How does our genomic intertwining with our microbial partners change how we understand relatedness or selfhood? What does it mean to define our states of health through our microbial selves? When thinking about this symposium, and especially this session topic, I realized that social and biological scientists may not even start with the same idea of social determinants or even health. For human microbial ecologists, the health that is important to studying the microbiome might consist of diet, geography, genes, mode of birth, vaccinations. As an anthropologist thinking about mothers and babies in Dhaka, all of those things matter, along with important influences like gender, class and ethnicity, working and environmental conditions like garment factories that exist in Dhaka, the violence of a colonial national history, corrupt or non-existent governmental systems of sanitation and water. We see that when we start to pile up all these concepts of health on top of each other, the picture becomes very messy and confusing. But it also becomes more detailed, more complete, and eventually, hopefully, more precise. This is why microbiome science and anthropology need each other. While I was in the Gordon Lab in 2010 and 2011, at least 6 lab members were working specifically on the gut microbes and malnutrition, and 3 were dealing directly with samples from Dhaka. The number of lab members, as well as those working on both malnutrition in Bangladesh, has substantially increased in subsequent years, becoming a central focus of the lab's work. My research focused on 2 projects, one on analyzing the Dhaka samples and the other conducting diet experiments using what they called Bangladeshi food and germ-free mice humanized with microbiota from Bangladeshi donors. I was specifically interested in these studies because they analyzed microbial material from babies in a specific geographic locale with a specifically defined health and nutritional status. These studies involved human beings I would be able to talk to at a node in the network I could travel to and observe. I wanted to find out where the feces came from, what the conditions were in the place they were produced, and for the people producing them. So this is one of the study families, 2 of the children that were, the baby was enrolled in the study. I just, as an aside, want to say, as I always try to point out when I present these slides, I'm really reluctant to share photos of the Bangladeshi people I took during my fieldwork. On one hand, I think it's really, really important that all of you, and especially biological scientists, see faces of human beings. Often we think of scientific study subjects as samples, right? Fecal samples, metagenomic reads, the data that their bodies produce. So it's really important to see the human being. On the other hand, there's a lot of race, class, and national privilege that I have in portraying people this way. And I really am resistant to be a part of any kind of exploitation of those people. So I still haven't figured it out. So I will show some pictures, but I don't feel comfortable about it. I was interested in how Bangladeshi diets were represented in mouse experiments and how dynamic human eating would work its way into the scientific practice or the ways it would be left out. For instance, Bangladeshi food has a lot of chilies, garlic, and other spices in it. Those things can't be given to mice, they will die. So the sorts of diets were very edited, right, in terms of representing what a Bangladeshi diet actually was. The birth cohort of Mirpur consisted of children whose mothers were enrolled while they were pregnant. These mothers received extensive prenatal care, education on healthy eating, breastfeeding, hygiene, and gave birth at health centers rather than at home, which is the local convention. The case control children were given macro and micronutrient supplements as well as therapeutic foods. All the families received free healthcare in exchange for their participation. This also is complicated and not value-free. The families I spoke to often felt like they were getting crappy healthcare because it was free and didn't always trust the doctor's advice. Each family was visited regularly by field research assistants who collected socioeconomic, surveillance, and food recall data. Sample collection and surveillance of cohort children began within 72 hours of birth. So meconium, the first stool of their lives, was entered into the study as their first sample that they submitted. Blood, urine, and fecal samples were collected regularly. So I think from the baby's birth to, I think through the first 3 years of life, sent to ICDDRB and then to the Gordon Lab for metagenomic analysis. During my time in Dhaka, 3 Bengali field research assistants introduced me to families in Mirpur enrolled in the Gordon Lab study. Children in their community suffered from extraordinary bouts of diarrheal illness, and malnutrition was often the result. The Gordon Lab was analyzing the microbial genomes of fecal samples from these babies to see what microbes they had and what the microbes were doing. I served as the lab ethnographer, gathering information on lifestyles, diets, and practices of everyday life. I want to give you a bit of an ethnographic snapshot here to think about how we can start to overlap the scientific and social concepts of health, how to use different kinds of expertise and data to do the most ethical and effective science possible. In this morning's session, folks were talking about populations and populations that are underrepresented in microbiome research. But the solution to this isn't just to sample from more diverse populations to have a wider representation. It's also to work with social science experts who are able to ask different kinds of questions, who can think carefully about who's going to benefit from the science. Where does the power lie? What are the social, political, economic conditions of life for people who we're studying? It's also critical to hear the voices of the people we're studying too. So this is Kanta and Rimi. These are all pseudonyms, so not their real names. Some of these families are still enrolled in studies, and a modicum of privacy is what I'm attempting. Kanta does not like eating. She's 18 months old and quick on bare feet out the open door of the nutrition center in the hot Dhaka street 3 times before her mother Rimi catches her. Typically gentle and affectionate with her daughter, Rimi holds Kanta firmly in the green light of the nutrition center, under illustrated posters teaching about nutritious food and hand washing. Kanta cries loudly as Rimi tries to spoon cereal into her mouth. Quote, she hates the pushti packets, but she doesn't like any food, so we force her to eat. Pushti packets are locally produced, ready-to-eat therapeutic foods, macronutrient meals containing rice powder, lentil powder, molasses, and soybean oil. The Bangla word pushti translates to nutrition in English. Although half of infants in Bangladesh weighed less than 5 pounds at birth, almost 2 pounds under WHO birth weight standards, it is when children get very sick that families seek help from ICDDRB, which has an influential historical presence in Mirpur. After local health workers diagnosed Kanta as malnourished, Rimi enrolled her in the microbiome discovery study. Quote, I thought my baby had a vitamin deficiency, and most importantly, I wanted medical care for her. It's very good for my baby because she was underweight and now she's in good health. I don't mind giving the feces and blood samples. They can use them to find if my baby has any diseases. They will know about it and they can treat it, end quote. ICDDRB is an international health research organization and also a free public hospital in Dhaka that serves hundreds of thousands of patients yearly. Credited with the discovery of oral rehydration therapy for the non-hospitalized, non-intravenous treatment of severe diarrhea, ICDRB is known locally as the cholera hospital. During the monsoon, or what is called cholera season, the ICDDRB serves hundreds a day, patients that spill out into the temporary cholera triage unit in the parking lot, far exceeding their 350 bed capacity. To give some context, Bangladesh has the highest population density in the world, 2,700 people per square mile. This is a map of the neighborhoods that I was working in. Each one of those little rectangles is a home. There's In what ICDDRB considers the ultra poor areas, the population density is 10 times greater, or about 30,000 people per square mile. There's a very high burden of diarrheal disease in these communities. One field doctor told me that children who suffer from the most severe chronic illness can experience up to 300 diarrheal incidents before age 3. Kanta's fecal samples collected by REMI are packed on ice, travel to the ICDDRB hospital, and are eventually flown back to the Gordon lab. There, they are inserted into the guts of germ-free mice, which are then fed a specifically lab-made representative Bangladeshi diet, bringing together biological and social representations of life in Dhaka. Kanta lives in Block D of Mirpur 11, in a 3-room house with tin walls. In one room, Kanta sleeps with her mother and father, and in the other, her grandmother, grandfather, and their 9-year-old son. The 3rd room is occupied by a renter and her young daughter. 8 people inhabit this 20 by 24 foot space, sharing the attached bathroom and narrow kitchen with 2 gas burners, a supply water pump, and pit latrine. Daily, government and NGO tankers truck water into the communities and fill underground tanks, which are connected to above ground pipes. Drinking water in Dhaka is notoriously and extraordinarily contaminated with sewage, agrochemicals, oil spillage, solid waste dumping, and untreated industrial effluent. While boiling can eliminate some contaminants, it does not rid it of high levels of arsenic and other heavy metals. In Kanta's house, there are no doors, no windows, no shoes. On her street, there are chickens and ducks underfoot and an open drain in front of every house. Rimi cooks food for the whole day before the gas service goes off at 9 a.m., preparing huge pots of rice, dal, spinach, and potato mash, and negotiating time at the stove with 5 other families who share it. The way in which Kanta's microbiome, her microbial self, comes into being is dependent on its social as well as its biological location. Along with birth, breastfeeding, and genes, excuse me, it's shaped by local practices surrounding water, chickens, and bare feet. Excuse me, I actually joked that this might happen during my talk, and now it's happening. The microbial populations in Kanta's gut are specific to the dilemmas that these bring into being. But how do we account for the social and environmental factors? We have yet to correlate or integrate ethnographic data about these social conditions into data analysis with metagenomic microbial genomes. Microbes that come from inside people's bodies are manipulated in the guts of mice as objects for understanding the structure of disease. Sorry. Why these specific Bangladeshi bodies are chosen for biomedical research and intervention brings into familiar oppositions, global and local, rich and poor, right? High income, low income, brown and white. Our biological selves come into being, function in the world, manifest bodily forms, and enact types of health and illness, all through the agency of microbes. If these microbes both affect and can be affected by who we love, where we live, and what we eat, if microbes make up our genetic and physical selves, aren't then our political, social, and economic selves also at least partly microbial? Malia, mother of 17-month-old daughter Priyanka, was receptive to the questions I asked, but sometimes would think them a bit too probing. Malia grew up in the house we sat in. She now lives there with her mother, husband, and 2 children. Her husband owned a small garment factory, and she embroidered so sorry, saris at night when the baby was asleep. She said, quote, "When I was a child, food was very tasty and fresh. Things like spinach don't taste good anymore, so my children eat less. These days, people use chemicals on plants like fertilizer, pesticides, and it makes food taste different. Soil has become very dirty and contaminated. In the past, there were small amounts of plastic, but now there are huge amounts of plastic contaminating the soil that the food is growing in," end quote. Like other mothers in the study that I talked to, Malia was very sensitive to changes since her childhood. She saw environmental contamination as an explanation for kids eating less and becoming malnourished. Packaged food is sold everywhere in the neighborhood and is expensive. She is acutely aware of her material and nutritional environments. She takes seriously the microbial education that the doctors have given her and her responsibility as a mother to protect her family from microbes. I'm so sorry. The FRAs, or field research assistants, teach the mothers the word jibanu, in Bangla, literally causative organism. Jibanu is not a word in everyday conversational Bengali, and not exactly the same as bacteria, microbe, or germ. For mothers, it became a concept bridging food, water, and places in the home to babies with diarrhea. This is an important point, as mothers were encouraged to think about themselves in new ways, about their bodies and their homes, their hands, their babies' mouths, and how their flesh touched pot handles, latrine buckets, and okra stalks. Mothers enrolled in the Microbiome Discovery Project began to think of themselves differently. They conceived of jibanu as part of places in their lives, like markets, street food vendor stalls, their own kitchens and latrines, but also thought of jibanu as part of themselves, as organisms they carried with them and passed on to their children. So gut microbes are studied and understood through their collective genomes and metabolic processes. Studies of human malnutrition vis-a-vis microbiomes are at risk for resulting in public health interventions that only focus on fixing the damaged or immature microbiome. Much of the Gordon Lab subsequent work has talked about probiotic and prebiotic interventions to fix an immature microbiome. But what are the ways in which we can account for the biology and social inequalities, like poor infrastructure, poverty? Can we see the social simultaneously and inextricable from the biological, not as metadata, but as crucial interacting data? Can we situate biomedical conditions in a socioeconomic context so we can design real-world action that cares for the whole self? This kind of collaborative work is critical, but also deeply difficult, frustrating, and sometimes painful. It contains imbalances of power, funding, and research goals. As I've talked about today, social determinants of health are refigured through a focus of microbes in Dhaka. Observations on the ways of living, caring for children, and feeding families, as well as larger analyses of structures of power, violence, race, and gender can help broaden the scientific view of study subjects. Sorry, I lost my place there. I paid careful attention to how the responsibility and compliance get assigned to specific bodies and thought about how to help reroute scientific and global health interventions down a more equitable path, one that accounts for the material, environmental, and social vulnerabilities that contribute to the making of microbiomes. After my work with the Gordon Lab, I have done subsequent work. There are a lot of really great interdisciplinary, no, I shouldn't say a lot. There are a few very good interdisciplinary working groups, such as the Microbes and Social Equity Group, that is bringing together experts in different disciplines to think about microbiomes in a biological and a social way. These are my field research. In 2021, some collaborators and I who were from all different fields, a microbiologist, a geographer, an evolutionary ecologist, and I wrote this paper about the future of an anti-racist microbiome science. This touches on a lot of what Dr. D'Amato was talking about in terms of how we think of race in these studies, and how we account for racism and how it impacts people, environments, and microbiomes. Excuse me. So this work was written for a scientific audience, but I feel like it has been eminently impactful and is one of the most meaningful things that I have worked on, because we're thinking about structural drivers of microbial populations and not just using race as a proxy to talk about different kinds of populations. And just the last thing I'll say is as a shameless plug is that a book that I wrote about all of my work with the Gordon Lab is coming out this spring from University of Minnesota Press. So just got into the catalog today. So that is my decade of work with the Gordon Lab. I want to thank the Gordon Lab and all of the subjects for allowing me to talk about their lives. Thank you.